So the first one which comes to mind is something called nor-ursodeoxycholic acid. Now, ursodeoxycholic acid is a sort of a bile acid that we give to patients with cholestasis, and it's what we call a choliretic, helps the bile flow better, and there are some studies which suggest it reduces cell death in the bile ducts, reduces apoptosis. And it has been shown to have benefit in other cholestatic liver diseases, but in PSC, it may improve liver blood tests to a certain degree, but because of the nature of the condition and the amount of fibrosis and scarring you have around the bile ducts, it doesn't really work very well. And nor-urso, however, is sort of it's like urso deluxe, which has got an extra molecule attached to it, and because of the way it undergoes circulation between the liver and the gut, it has an increased potency and an increased mechanism of action. And certainly in preclinical animal models, it's been shown to improve liver injury, including liver fibrosis, and that's currently undergoing clinical trial. And our centre is part of a multi-centre effort in that regard. And the other one is a study by Gilead, which is targeting a molecule known as LOXL2, which is a pro-fibrogenic molecule within the liver, and and the endpoints for that study, unlike the nor-urso, which relies on liver biochemistry, the LOXL2 study is looking at liver fibrosis. So individuals over a 2-year period will receive the drug or sort of placebo, and will have 3 liver biopsies, beginning, middle and end, to see if fibrosis is resolved with this therapy. But one of the molecules that I'm interested in is going to be the subject of an emerging clinical trial, of which we hope to recruit the first patient sort of in the next few weeks, is a molecule known as vascular adhesion protein 1, or VAP1, which is a key player in regulating the expression of other adhesion molecules within the liver, as well as being an adhesion molecule itself. And it has unique properties as an enzyme which sort of drive liver fibrosis, and one of the talks that I'm giving on the Friday is surrounding VAP1 in PSC. VAP1 actually controls LOXL2 activity. That's something that's sort of beyond the scope of discussion today, perhaps, but but as well as controlling the expression of these other molecules that I talked about. So that is sort of a trial that's due to be open in the next couple of weeks. The third sort of trial, which is due to open, targets a molecule which I mentioned earlier called MADCAM1, which is expressed in the liver and in the gut of patients with inflammatory bowel disease. Vedolizumab targets the ligand for VAP1, and the ligand is alpha-4-beta-7, and there are a wealth of emerging publications in high-impact journals looking at success in colitis and inflammatory bowel disease, and given the link with PSC and inflammatory bowel disease, vedolizumab is now moving into PSC clinical trials. And those are the sort of the big four that are sort of we were likely to see some activity in 2015. There's another report on the meeting. They're still actively recruiting patients. In fact, they're still at the active recruitment stage. So mouse studies for nor-urso have been reported, and they show that improvement in bile acid flow, improvement in liver biochemistry, and improvement in liver fibrosis. So that's certainly the one that's been studied the most, and that's the one that's probably the most far ahead in its development. Vedolizumab has been reported in colitis patients, but not in models of animal liver injury. And that's perhaps because the molecular expression of some of these molecules in mouse models of liver injury are very different. So you don't see MADCAM1 or VAP1 being expressed in the liver of mouse models of, that have liver injury. So it's very different. So I think mouse models have only taken us so far. I can only answer certain questions, and I don't think even targeting VAP1 or MADCAM1 in the existing models that we have in mice would likely to be fruitful for that reason.